Hello, nice to see you again. In this video, we'll be continuing learning about the brain and looking closer to the cerebral cortex as the most prominent part of the mammalian brain. All right, so as you can see in these pictures here, so the basic organization of the cerebral cortex is a remarkably similar across vertebrate species. So the comparison here is some of the brain of mammalian here. And so, for example, those organizations are the visual cortex is in the same place amongst mammalian brains, and the auditory cortex is also in the same place among mammalian brains, and so forth. However, even though the organization is almost similar, but the brains vary enormously in size. So the largest mammalian brains are 100,000 times larger than the smallest one. For example, if we compare mammalian species, we see the differences in size of the cerebral cortex and the degree of the folding, right? And compared to other mammals of comparable size, such as the primate, the monkey, the apes, and the humans, they have larger cerebral cortex, and they have more folding as well, and more neurons per unit of volume. Compared to larger animals such as elephants, yes, elephants have larger brain size, but also larger neuron, but fewer neurons per unit of volumes. And humans have almost 3 times as many neurons in the cerebral cortex as elephants have, although the elephant brain is more than twice as large. Interesting, right? All right, so let's take a look closer on the cerebral cortex. The cells on the outer surface of cerebral cortex here are the gray matter with the darkest color here, and their axon is extending inward are the white matter here, the lighter color, and neuron in each hemisphere communicate with neuron in the corresponding part of the other hemisphere through 2 bundles of axon, the corpus callosum and the smaller anterior commissure. So several other commissure or the pathway across the midline are linked to subcortical structure. And the most fundamental part of the mammalian brain and consists of cellular layers and on the outer surface of the cerebral hemisphere. And the cerebral cortex is divided into 2, the left and the right? And is connected by 2 sets of axon called corpus callosum and the anterior commissura here. And the human cerebral cortex is highly developed compared to other species. And the microscopic structure of the cells of the cerebral cortex varies from one cortical area to another, as does as the density of the neurons per volume. So much research has been directed toward understanding the relationship between structure and function. In human and most other mammals, the cerebral cortex here contains up to 6 laminae or 6 layers of the cell's body that are parallel to the surface of the cortex and separated from each other by layers of fiber, as you can see in this figure. So the laminae vary in thickness and prominence from one another, from one part of the cortex to another. And a given laminae may be absent from certain areas. So for example, laminae 5, which sends long axons to the spinal cord and other distant areas, is thickest in the motor cortex, which has the greatest control of the muscles, right? But lamina 4, which receive axon from the sensory nuclei of the thalamus, is prominent in the sensory areas of the cortex, which are the visual, auditory, and somatosensory, but absent from the motor cortex. And the cells of the cortex are also organized into columns of cells perpendicular to the laminae. So in this figure, you can see that they're illustrating the idea of the column, although in nature, they are not so straight like that. So the cells within a given column have a similar properties to one another. For example, if one cell in a column responds to touch on the palm of the left hand, then the other cells in that column do too. And another example, if one cell responds to horizontal pattern of light at a particular location, then other cells in the column respond to the same pattern in nearby location as well. All right, we now turn to the specific part of the cortex. So researchers make fine distinction amongst area of the cerebral cortex based on the structure and function of the cells. And for convenience, the researchers grouped these areas into 4 lobes named for the skull bones that lie over them. There are occipital, parietal, temporal, and frontal lobes. We will see the first one, the occipital lobe. It's located at the posterior or caudal end of the cortex. Yeah, it's in the back side of your brain. Yeah, it's the main target for visual information. And the posterior pole of the occipital lobe is known as the primary visual cortex or the striate cortex because of its striped appearance in cross-section. Yeah. So the destruction of any part of the striate cortex causes cortical blindness in the related part of the visual field. So for example, extensive damage to the striate cortex of the right hemisphere causes blindness in the left visual field. That is the left side of the world from the viewer's perspective, right? So the person with the cortical blindness has normal eyes, but no conscious visual perception and no visual imagery, not even in dream. So people who suffer eye damage become blind, but if they have an intact occipital cortex and previous visual experiences, they can still imagine visual scenes and still can have visual dream as well. In short, the eyes provide the stimulus, but the visual cortex provides the experience. All right, we move on to parietal lobes. It lies between the occipital lobe and the central sulcus, a deep groove in the surface of the cortex. It's in the top of your head, yeah, like here. And the essential, it is essential for spatial information, numerical, spatial awareness, and spatial perception, or the proprioception. Yeah, it's awareness of body parts in space and in relation to each other. So the area just posterior to the central sulcus, the postcentral gyrus, or the primary somatosensory cortex, they receive sensation from touch receptors, from the muscles receptors, and joint receptors. We will take a look closer here. The postcentral gyrus included 4 bands of cell parallel to the central sulcus. Its separate areas along each band will receive simultaneous information from different parts of the body. As you can see in this figure, 2 of the bands receive mostly light touch information, 1 receive deep pressure information, and 1 receive combination of both. In effect, the postcentral gyrus here represent the body 4 times. And information about touch and body location is important not only for its own shape, but also for interpreting visual and auditory information. So if, for example, if you see something in the upper left portion of your visual field, your brain needs to know which direction your eyes are turned, right? So the position of your head and the tilt of your body is important before it can determine the location of whatever you see. And the parietal lobes here monitors all the information about the eye, the head, the body position, and then passes it on to the brain areas that control movement. So the parietal lobe is also essential not only for spatial information, but also numerical information, as I said before. So that overlap makes sense when you consider all the ways in which numbers relate to the space. So including the fact that we initially use our finger to count, right? All right, the next area is the temporal lobe, is the lateral portion of each hemisphere near the temples here. Yeah, it is primary cortical target for auditory information. So the human temporal lobe, in most cases, the left temporal lobe is essential for understanding spoken language. And the temporal lobes also contributes to complex aspects of vision, including perception of movement and recognition of faces. So a tumor in temporal lobe may give rise to elaborate auditory or a visual hallucination, whereas a tumor in the occipital lobe ordinarily evokes only simple sensations such as flashes of light. All right, we explore this issue further in the chapter of emotion later in Biopsychology 2 class. Following that, the last one is the frontal lobe, it containing the primary motor cortex and the prefrontal cortex. It extends from the central sulcus to the anterior limit of the brain. Yeah, it's here, located here. And the posterior portion of the frontal lobe, the precentral gyrus, is specialized for the control of fine movement, such as moving your fingers. And the most anterior portion of the frontal lobe is the prefrontal cortex. So in general, species with a larger cerebral cortex devote a larger percentage of it to the prefrontal cortex. So for example, it formed a larger portion of the cortex in humans and the great apes than in other species. Okay, so neurons in the prefrontal cortex have huge numbers of synapses and integrate an enormous amount of information. So it is very important. An analysis of thousands of studies concluded that the prefrontal cortex has three major regions. So the first one is the posterior portion is associated with mostly with movement. The middle zone pertains to working memory, cognitive control, and emotional reaction. So working memory is the ability to remember recent events, such as where you park your car or what you were talking about before an interruption, and etc. And people with damage to the prefrontal cortex have trouble on the delayed response tasks, in which they see or hear something, and then they have to respond to it after a delay. Okay, so they have difficulty in that. And afterwards, the anterior zone of the prefrontal cortex is important for making decisions, evaluating which of several courses of action is likely to achieve the best outcome. Yeah. So when you decide whether to do something, you consider the difficulty of the action, right? The probabilities of success or failure, right? And how valuable the possible reward would be to you, and all things that considered before deciding something. All right. So cells in the prefrontal cortex respond to all these complex factors. So people with prefrontal cortical damage often make decisions that seem impulsive because they fail to weigh all the likely pros and cons before deciding something. And prefrontal lobotomy, or the damage in the prefrontal cortex or the disconnection, will connect to apathy, memory problem, and lacking of emotional expression. Later on, how do the brain parts work together then? Okay, so here's a theoretical issue that researchers hardly even considered before about 1990. So how do various brains are combined to produce a unified experience? So when you eat something, you experience the smell in the nose and the taste, as well as the touch or on the tongue of the food that you eat, right? And all become a single experience of eating. So another example, if you shake something that makes a noise, you perceive that that what you see is also what you feel and what you hear, right? But how do you do that? Each of that senses activate different areas of the brain, right? A different area of the cortex. And those areas have only weak connection with one another. All right, so the question of how various brain's area produce a perception of a single object is known as the binding problem, or the large scale of integration problem. In earlier era, researchers thought that various kinds of sensory information converge onto what they call the association areas of the cortex. Their guess was that those areas associated one sensation with another, or current sensation with memories of previous experiences. But later, research found that relatively few cells combines one sense with another. Even when they do, they don't fully answer the question of how we bind sensory information together. So for example here, certain neurons in the posterior temporal cortex, both when you see a chainsaw or when you hear the sound it makes, or both when you see a jackhammer and when you hear a jackhammer sound. But surely we weren't born knowing what sound a chainsaw or a jackhammers make, right? Somehow those cells had to develop those properties through experience. Similarly, many neurons in the superior collicus respond to more than one sensory system, but they constantly change their property based on experience. All right, so although researchers cannot fully explain binding, they know what is necessary for it to occur. It occurs if you perceive two sensations as happening at the same time and it approximate the same place as well. For example, here, you can position yourself parallel to a large mirror in front of you, as you can see in this figure, so that you can see your right hand and its reflection in the mirror. All right, so keep your left hand out of the side, and now repeatedly clench and unclench both hands in unison. Wiggle your fingers, right? Touch your thumb to each finger and so forth. In each case, doing the same thing with both hands at the same time, right? You will continually feel your left hand doing the same thing you see the hand in the mirror doing, which being the mirror image of your right hand. So it looks like it will look like it's your left hand. Okay, so after 2 or 3 minutes, you may start to feel that the hand in the mirror is your own left hand. So the evidence then indicates that we bind to experiences that occur at the same time. Still, the theoretical questions remained of exactly how we do so. All right. Applying these principles, then researchers arranged a camera to video someone back and simultaneously send the picture to a 3-dimensional display mounted to the person's head. As you can see in this figure, imagine that you are the participant as you view the video of your back. It appears to be 2 meters in front of you. Then someone stroke your back. You simultaneously feel the touch and see the action that appears to be 2 meters in front of you. After a while, you start perceiving your body as being 2 meters in front of you. When you are asked, please return to your seat, you will walk to a spot displaced from the actual seat as if you were actually 2 meters forward from your current position. It's interesting, right? Suppose you see a light flash once while you hear 2 beeps, you will sometimes think you saw the light flash twice. If the tone is soft, you may experience the opposite. The tones may beep twice during the one flash of light, and you think you heard only 1 beep, okay? So if you saw 3 flashes of light, you may think you heard 3 beeps as well. That's another example of binding problems. So bindings here often fails if the display are flashed very briefly or while the viewer is distracted. That's a very interesting findings of research about bindings illustration. All right, so that's all about the brain, and thank you for your attention. We'll see you again in the next topic. Thank you, bye-bye, and take care.